Now we are going to begin our discussion of RNA sequencing. This video will serve as an introduction to RNA sequencing, explaining what it is, what we can use it for, as well as bring to light important considerations for selecting library protocols to meet the specific needs of your experiments. Furthermore, we will discuss the difference between steady-state and nascent RNA sequencing. What is RNA sequencing? RNA sequencing is a high-throughput, sequencing-based method for transcriptome profiling. While all the methods thus far introduced in this workshop have involved sequencing fragments of DNA from the genome, RNA-seq instead sequences fragments of RNA from the transcriptome. The transcriptome refers to all the RNAs of a cell or organism. This includes mRNAs, which go on to be translated into proteins, as well as various sorts of non-coding RNAs, ribosomal RNAs, transfer RNAs, etc. RNA-seq can be used to address a number of questions, but the primary use of RNA sequencing has been to catalog and quantify gene expression. This information can be used to determine tissue-specific or developmental-specific expression patterns. It can be used to detect and monitor disease phenotypes, but most commonly, quantification of gene expression by RNA-seq is used to perform differential expression analysis, which seeks to identify genes whose expression is significantly different between two or more samples. RNA-seq has also been an immensely powerful tool for novel transcript discovery and allows for transcriptome sequence assembly without the need for a reference genome sequence. There are many different libraries available for making RNA-seq libraries, and it is important to consider the goals of your experiment and the specific questions that you wish to address when selecting an appropriate protocol. For instance, the type or types of RNAs that you wish to sequence will determine how you prep your RNA before library construction. If you are interested in sequencing small RNAs, like microRNAs, there are specialized protocols for doing so. Likewise, it is very useful to either enrich for polyadenylated RNAs or to perform a ribosomal RNA subtraction. Otherwise, as much as 80% of your RNA-seq reads will be ribosomal RNA in origin, and you will get very poor sequencing efficiency and low coverage of your target transcripts. Additional considerations for selecting library protocols depends on the type of questions being addressed with RNA-seq. For instance, spike and controls are commonly included in library preps to improve the accuracy of transcript quantification. Likewise, longer read lengths and paired end libraries are recommended if you are interested in de novo transcript discovery and differential isoform usage and expression. An additional and important consideration for RNA sequencing is that RNA-seq only measures steady-state RNA levels and is not a direct readout of gene expression or transcription. In the cell, there are two opposing pathways that contribute to the level of any given RNA. Transcription adds RNA to the cell, and degradation removes it. The difference in the rate at which an RNA is made and the rate at which it is degraded ultimately determines how much of that RNA will be found in the cell at any given point in time. Changes in gene expression observed by RNA sequencing cannot distinguish between changes in the rate of transcription and the rate of degradation. For instance, when RNA levels increase in the steady-state pool, that is an indication that transcription is occurring at a rate faster than degradation. But whether that is due to an increase in transcription or a decrease in degradation is unclear. The same issue applies when RNA levels decrease. Although this is an indication that degradation is occurring faster than transcription, it again is unclear whether or not this is due to a change in transcription or degradation. So if you are interested in measuring active, nascent transcription and not just steady-state RNA levels, then you need to use a protocol other than RNA sequencing. The most popular method for nascent RNA sequencing is global run-on sequencing, also known as GrowSeq. An alternative approach is to use nascent elongating transcript sequencing, which is known as NetSeq. One thing to note about NetSeq is that there are different protocols for mammalian NetSeq and the original NetSeq, which was originally developed in yeast. If you will be using one of these methods in your experiments, then you will need to use a different analysis pipeline than what will be discussed in this workshop for RNA sequencing.